Hello.
Hello, good morning.
Good morning. Do I need to show myself?
No, you're okay.
Okay.
We'll get started in a couple minutes. I'm sure there's going to be a couple people joining, like 11:32-ish.
Okay.
But if I could have your name and your student ID number, that would be perfect for me. I just need to sign you guys in.
Do you want it in the chat box or do you want it?
Yeah, in the chat box, please. Perfect. Thank you. Alrighty. Hello, Adrian.
Hey.
Thank you. Perfect. Okay. Well, I think I'm going to go ahead and get started. And if anybody else joins, then we'll just recap if need be. But I saw you guys last night. Or I saw Adrian last night. There's only two other people here. So there isn't like anything new other than the lecture material this morning. So how are we feeling on that?
A little bit better since I actually reviewed the other day, so I'm preparing to this lecture.
Okay. Oh, good. That sounds good. Okay. Is there any questions we have about the lecture material?
Today's lecture just seemed like it was quite a bit.
Yeah. They're gonna get a little bit more dense as time goes on. That's why it's kind of like how I was saying in a couple other meetings, like I would write out what would be on the PowerPoint and then I'd go to lecture. That way I can follow along a little easier.
Okay, so you're suggesting reviewing the PowerPoints before she lectures.
Yes.
Okay.
You don't necessarily need to teach yourself. It's just maybe if you like to take notes on it, you write down whatever you'd like to take from the lecture notes or from the PowerPoint. So when you go into lecture, you can take notes on it. And if something doesn't click and you're looking at it, you can ask questions and you're not like, I need to write this slide down, but I'm already 3 slides back and I need to ask this question. And I don't know what's going on.
Mm-hmm.
So that's what I used to do. And it helped me quite a bit.
Okay. The way I'm currently doing is I'm listening to her lecture and all my notes are going directly on to the PowerPoint.
Mm-hmm.
And I'm only referencing the book if I need a little bit more clarification.
Yeah, that's a pretty good way.
In her syllabus, it's saying to read the chapter. However, I haven't read a complete chapter.
You really don't need to. I only did it for the first couple chapters, and it's more of a fluff time waster type of thing.
Mhm.
It's not really anything that you can gather a whole lot of information from. There might be a few good visuals that you can pull from the book, but a lot of the same visuals are used in the PowerPoint notes.
Okay. Would you be able to explain gram positive and gram negative?
Yeah, yeah, of course. So let me pull up a quick reference for me so I can kind of draw it out, help you guys visualize it, and describe it as I'm going.
Okay.
Before we dive too deep, I'm also going to address a couple other things. So in terms of anything in person, I'll repeat this a few times. I'm sorry if it sounds redundant, but in terms of anything in person, in terms of like PAL sessions, unfortunately, we don't have the AOK yet, and they're going to try to reevaluate in March. That's a long ways away, but it's going to be closer to when the content gets heavier. So let's cross our fingers and hope that maybe we can do something in person.
Okay.
And something like that. But I had a question a little while ago. I just wanted to see if there's anything like that. Also, this green part of the marker just wouldn't erase, so now upset. I don't have a big eraser either. And I have different colors, so I even have purple. So right. So you want gram positive over here and gram negative. Let's separate this out. So
I can't really see that.
is it too light?
Yeah.
Okay, we'll go over it red. This is chisel. Better?
Yes.
Okay. I'll use red for now. If there's anything purple, just color it in kind of heavy. So, pull a little. I'm trying to go as close to the lecture material as possible so that I don't bring anything new in. Let's see. Okay. So when we're thinking about a gram negative bacteria, we're going to have 2 phospholipid bilayers and 1 thin peptidoglycan layer, a periplasmic space, and then that second phospholipid bilayer. So what that's going to look like on our gram negative side is we're going to have, right, this is our phospholipids. Phospholipid bilayer going on. Right.
Okay.
And then next in line, oops, goes on. Next is going to be that thin peptidoglycan. Then we're going to have a bit of a space. It's basically like negative space. And then we're going to have a second phospholipid bilayer. Okay. So this is what a gram negative is going to look like. They don't have a whole lot of peptidoglycan, and they especially don't have teichoic acid. So that is what I'm going to put on the slide. I hope you can see that little. So that's what our gram negatives look like. Now, to contrast to our gram positive, I'm going to move my chair a little bit. Okay. Gram positive is going to look like a big swath of peptidoglycans. So this is our thick peptidoglycan layer and our gram positive, right? Also sprinkled about, these are going to be your teichoic acids. Sorry, that looks like it. There you go. So gram positive teichoic acid, right? Thick peptidoglycan layer. Then we have the same paraplasmic space here. I'll use that again. So then we have our, I'm probably going off a little bit, but then we have our space here and another phospholipid right there. The only difference between gram positive and gram negative, or the most important, I should say, are their walls. So these are their cell walls, and that is the primary difference between the two.
So is it safe to say that you will see gram positive on prokaryotes because they have the peptidoglycan? Peptidoglycan, yes.
So both of these we will find in bacteria. We primarily bacteria. These are how we differentiate bacteria.
Oh, gram positive and gram negative.
Yes, it's one of the first tests you'll do. It helps you see on a microscopic level what a bacteria looks like. I know you guys talked about cocci, rods. So later on in the semester, you'll learn, okay, well, if my bacteria looks like this, and let's say it was like a deep dark purple, that's gram positive, and it's a rod. So you're gonna label it as a gram positive rod. See how that works?
Uh-huh.
It's just a way to differentiate bacteria, and it's a really foundational thing. That's why we're covering it first. We're not going to go into something super niche right off the bat.
So if it's lightish pink, it's mostly going to be gram negative.
Yes.
Is that with any bacteria?
If this is how their wall, if it's either this or this, and that's what their walls look like, yes. There are probably some weird bacteria out there that present as one thing, but are actually another. Because you'll learn today, and I can go over it again on Monday and review it, but you guys will learn today. And if you were on Wednesday, I don't know if you've done the gram stain yet, but when you do the gram stain, you'll learn that there are different processes that will help that differentiate this. So because there's a thick peptidoglycan layer, we put this dye in and it stays here, right? But since there's not a whole lot here, we put this dye in and then only some gets absorbed, but it gets washed out. So it appears pink. So with these differences, you'll kind of see how they work in the lab.
Okay.
I don't want to go into it too much just because I don't want to confuse you guys right off the bat. But know that, what they physically look like at this level and what they look like under a microscope, they correlate. Let's see. So let me also note on here That, because I also want to get back to like the lecture material so I don't confuse you guys. So this is considered the outer membrane. I'm just going to label it mem because I'm probably going to misspell it somehow. So this is our outer membrane. We jump right to the peptidoglycan here. So there's no outer membrane here for the gram-positive. For the gram-positive as well, there is kind of a space, but not really. It's not a defined space right here. But this is like the actual space that they have. On the pictures, it looks like there's like a really definite space, but technically there's no paraplasmic space. Gram-negative has a paraplasmic space, gram-positive does not. Let's see. Trying to think. Is there anything? Did I confuse you guys on this? I hope I didn't.
No, that
No.
helped a lot.
That helped, yeah.
Okay, good. Let me see. So one thing that we will also find, so we'll learn too that their walls are made up of different substances depending on what their structure is. So we have an outer membrane here on our gram-negative, and that means it also has the LPS. Do you guys remember what that stands for?
No.
The lipopolysaccharide. Now this is important because it's going to be a pathogenic characteristic. LPS is characteristic of endotoxins. So let's look at those. Let's look at that word, right? Endotoxin. So endo meaning inside, toxin. So if a bacteria is coming in and releasing an endotoxin, it's going to do damage. So we can see that we have pretty pathogenic bacteria in the gram-negative side, and that would be a characteristic of it, right? So not only do we need to treat the bacteria and kill the bacteria in this scenario, we also need to treat the consequences of the toxin and the effects of the toxin that has on the person, right?
Yeah, I want to say she talked about that this morning. Yeah.
Yeah.
She did.
It might have been brief, but I'm going to tell you right now, it's going to be on the exam and it's going to be in the future. You'll build on this concept of the LPS.
Okay.
Do you think it will be like a short answer type of question, like how LPS will destroy or affect the body?
It could very well be. That is a good. I don't know. I might have to tell her that would be a good one, but
No, it's okay.
it, definitely. It's like, so what's the process? This bacteria is in your body and it has an endotoxin, and if we start killing the bacteria, the endotoxin is getting released. So this endotoxin causes a slew of physiological processes. You're going to have vasodilation and drop in blood pressure, and that both of those things not good, because you're just basically going to go into shock. So you're going to probably be like, oh well, this is just a process of the infection, right? You're going to give more antibiotics, you're going to kill more bacteria, and the process starts again. You're going to release more of that LPS once those bacteria die, and you're going to continually dilate those blood vessels and drop the blood pressure, and that can lead to death. That is definitely what you want to be careful of in the healthcare field. It's potentially very fatal, and it can go, you're scratching your head like, what's happening? This person has an infection and they're dying. And it's like it was like E. coli, which a lot of people recover from. It just depends. But E. coli is also, yes, gram negative. You'll learn about that as well. You'll test several different ones, you'll plate them. That's all going to be next week. You'll get a little bit more in depth and get more comfortable with plating and the gram stain.
Can I rephrase that real quick? So basically, gram negative stain bacteria, they're dangerous because in their cell walls, right? They have the LPS, the lipopolysaccharide.
Yep.
And then LPS, they have that lipid A, which is toxic. So
Mhm.
therefore, if the doctor will prescribe antibiotics to someone who has gram negative, the lipid A will be released to the blood vessels, which was cause it to dilate. And then they think it's working, but then the more they give antibiotics, the more lipid A will be produced, right?
Yes. So
And
that's why. So
the-
I'm sorry, go on.
And that's what's causing the vasodilation, the lipid A, or
Yes. The
Oh.
release of the endotoxin, yes.
which causes shock, which would cause bad things, basically.
Yes.
I
Shock
see.
is always going to be bad.
Thank-
But yes, right on the dot. You hit the nail on the head. So I'm trying to think. I was gonna say something about that. Oh, the thing is, like, we can't just be like, well, you have a bacteria, you're gonna have to figure out how to fight it off yourself. No, we're gonna treat you, right? If you walk into the hospital and you have an infection. So what we would do is we wouldn't overwhelm your body with antibiotics and start killing off a large bacterial load at a time, because there is a certain amount that your body can tolerate. Because think about like working out, right? You're changing your physiological blood pressure depending on what you do. Even standing up changes your blood pressure, right? So we can't always stay at our resting blood pressure, and slight changes are okay, and our body can adapt to that and maintain the homeostasis. So we give little bits of antibiotic to kill off bacterial load, and then it gives our body time to kind of process it, and it won't kill us. But if we start prescribing heavy antibiotics, we're gonna potentially harm the patient. But yes, you guys are good on the LPS. Sounds like you guys understand that in terms of the gram negative.
Yes. That was a good explanation. Thank you.
Yeah, no problem. I really appreciate you being like, oh, can I just repeat this back? Because that is a study tool. If you need to tell yourself in the mirror, this is what LPS does, do it, because it helps a lot. Let's see. What else could I? Oh, okay. So, like I said, with the bacteria isn't always black and white. I believe in the lecture she mentioned acid-fast bacteria. Does that sound familiar?
I think that we haven't gone through that.
Not yet. Okay. Well, you know how it's like not all the time bacteria can be tricky, and there's always the exception. That's the exception, is acid fast. In what their cell wall looks like. And they, don't quote me, but I want to say that they stain pink, and but they're built differently than gram negative, and they actually have a thicker peptidoglycan than gram negative. So when you guys go over that, we'll go over that together if you'd like. There's also bacteria that don't have a cell wall. That's another exception. I promise it's better than this. There's not always the exception, and I know it's like, okay, here's the rule, except this, and this. so it gets confusing, but I'm sorry. Let me think. We know about NAGNAM.
Yeah, she mentioned that in lecture today.
Good, good. So where's NAGNAM located?
I have to look at my notes.
The P, the P word.
The peptidoglycan?
Glycan.
Yeah, peptidoglycan is where our NAGNAM is. I always like to think about it, and I only wrote NAGNAM in purple so I'd remember that purple meant peptidoglycan. This big purple, little purple, that's
It's
where the
solely-
NAGNAM is.
It's solely made out of NAGNAM because I saw glucose in there too. I don't know if
That was a comparison between the two. So it's a hexose sugar. Glucose is hexose sugar, and the NAGNAM are also that 6-carbon ring. I think she was just comparing it.
Yeah,
But
she was just
it's
comparing
not
it to
necessarily
glucose.
sugar. There's a lot of special names for it. Or there is a special name for it, as in like a long, like, what is it? Nitrogenous acetylglucosamine, and then nitrogenous acetyluramic acid. I want to say that, but it's just the backbone. There are other things in peptidoglycan that make it peptidoglycan. It's just that's the backbone. Think of it almost like DNA. So you're going to have your subpart, you're gonna have your backbone, and that is glucose. Although in DNA, I'll give you it's a pentose sugar, not a 6-carbon sugar. So you'll have your backbones, and then you'll have things in between that cross-link with a peptide bond. Did you want me to draw that out for you guys?
Sure.
Okay. Okay, let me erase this. You know what, I'm just gonna, I have my little Lysol wipes, so I think this will help me on the board at least. You'll see. Yeah. Oh, yeah. Okay. Okay. Yeah, the green's going on there too. Because I'm just like this. I hate when there's residue.
quick question. Do you think on a possible matching question would be like name the parts of the microscope?
Yes.
Yeah, for sure. And like the name the parts of a cell. Like no.
A what?
Like the parts of a eukaryote or
Oh yeah.
yeah, possibly this as
Yeah.
well. Okay.
That's why I say if there's a diagram, always redraw it, because it's always good study practice. And sometimes you surprise yourself. You're like, wait a minute, do I know this? And then you're like making your best guess off of what you remember, and it's right. You know? All right. So let me draw this out for you guys. I'll do it in purple and I'll just go over it. Because I want you guys to think about the, purple means peptidoglycan. So this is what peptidoglycan looks like. So we have our NAM, right, and I'm pretty sure that it's not really like a special sequence, but I'm just gonna alternate it. I hope I left myself enough room. So from one NAM, we're gonna have a L-adenine. Let me use another color. It's gonna be L-alanine, Okay. And then we're gonna have glutamic acid. Okay. And then we'll also have I don't think you'll need to know this granular, but it'll be a diamino, wow, medic, something like that. I'm just gonna put DA. It's gonna be an acid. This is in your lecture notes. I'm just trying to redraw it for you guys. And then you'll have alanine at the bottom. Right? And then this is your little peptide cross link. And then I kind of didn't leave myself enough space, but these are the links between NAMs. So, and then it's just this copied here, and you're going to link those two together. And these are considered tetrapeptides. Okay. So we have our sugar backbone, our tetrapeptides, and together we have a peptide crossing. I don't think that you'll have to know the specifics of what is in here, but then again, she could be changing some of her material. She could be wanting you guys to know it, but I'll double check with her in class today. So I'll ask her to what extent do you guys need to know this. But yeah, that's what NAG-NAM. NAG-NAM is the backbone. We have this in between, and that's everything that's in peptidoglycan.
Okay.
Is there anything else that you guys wanted me to go over about structure, anything like that?
About like the main differences of prokaryotes and eukaryotes and how like your tips of remembering them.
Okay, yeah. So
And part
eukaryotes, think about you and me, right? One of their main differences is that they have membrane-bound organelles. You know, they have they're a little more complex. They have a nucleus with instead of a nucleoid space that's in prokaryotes. Let's see, what's some other differences? Oh, they don't have cell walls. Eukaryotes don't have cell walls. You and me, eukaryote, we're a little more advanced than that. We don't need those types of walls. So with a prokaryote, we're going to have the cell wall and outer membrane, and we're going to have on the inside, we're going to have ribosomes. We're going to have a nucleoid space with the DNA, and you'll learn in the future there's going to be a plasmid. You'll just need to draw a circle and make sure you know that plasmid is in prokaryotes. They usually have flagellum. We don't have flagellum, right? Our skin cells don't have flagellum. We have cilia on some of our cells, but we don't have flagellum.
Okay, I just wanted to point out a couple of things. So my office is right here.
So when she's talking about bacteria,
And
she's talking about prokaryote.
Yes,
Yeah.
bacteria, prokaryotes. There are
Prokaryotes.
eukaryotic parasites, but most of the time, bacteria is prokaryotic.
Okay. Okay. Thank you very much.
Yeah, no problem. If you guys are ready to go, I am too. I know lunch is right around the corner.
Yes. Thank
Well,
you.
yeah, no problem. Have a good one, guys.
Thank you, you as well.